of the shape of cells. And you've already noticed this when you've been looking at things under the microscope. So the names that we give the shapes are coccus if it's spherical, rod if it's rod-shaped, and then spirillum is often given, that's the term given if they have some kind of corkscrew shape. And it can be rather loose spiral, or it can be very corkscrewed like a spirochete. And so probably most of what you're gonna see under the microscope are gonna be coccus and rod. And then there'll be a few filamentous when we get into, of course, the fungi. And that's primarily because those microbes that we want you to get experience with, especially when we get into the physiology section, where you look at how they do their physiology. Most of the microbes that we pick for that are just tend to be coccus or rod shaped. But if you were to go out hunting for microbes in their natural setting, you would see more diverse shapes. And so, for example, you might have some that are stalked, where they have like this appendage that they can use often to stabilize and attach to a surface. And so very often, if you are scraping a log or a rock in a stream or river and look at it under the microscope, you might see a lot of appendage bacteria that are using some kind of stalk to attach to the rock or the log so that they can stay put even in a faster current. You're gonna see some yeast, for example. Sometimes you can cook a yeast that is normally unicellular and fairly coccus shaped to start growing out a hyphae. So we'll talk a little bit more about that when we get into yeast, which is a type of fungi. And then the fungi that I mean, the bacteria that I work with, Streptomyces bacteria, they are filamentous. They look like petite fungi under the microscope. So, all right, now let's think a little bit about their cell membrane. So this is a refresher. My assumption is that you've had training on cell membranes. So this is just a refresher. So you have your phosphate group attached to a glycerol. And then off of the glycerol, you have two fatty acids dangling off there. And then if you think about a cell membrane, what we have is this bilayer lipid membrane, which is being shown here in figure B, where you have the hydrophilic, they color coded it, so the blue portion, the blue balls here, that's hydrophilic, likes to interact with water. The fatty acid tails do not like to interact with water because they're hydrophobic. So if you dump some fatty acids and, you know, with the phosphate and the glycerol and the fatty acid tails, if you dump that into water, they're going to naturally form this bilayer because the fatty acids want to hide away from water, whereas the phosphate glycerol, they want to interact with water. And so it gives that lovely bilayer membrane. And so bacteria have that, just like we have this idea of a bilayer membrane that composes our cell perimeter. But then, of course, just like with us, they also have other things in that bilayer membrane, primarily a lot of proteins. And so here you're seeing a mock-up of different types of proteins. Sometimes you can have proteins that will span the entire width, transmembrane proteins. Sometimes they're only on the inner portion of the membrane or maybe the outside portion of the membrane. And so it just, you know, depends what microbe you're looking at, what habitat they're in, what kind of proteins they're going to have in their membrane. So in general, when you look at a microbe, their cytoplasmic membrane is about 6 to 8 nanometers wide. And certainly have a lot of embedded proteins. And they say that bilayer membranes are somewhat fluid. Now, here's another picture showing a mock-up of bacterial membranes. We'll talk a little bit more about some of these things that you're seeing, including the haploids. Okay, we've already talked about ATPases. All right, so here I'm just going to point out, you see these haploids that are interspersed in the hydrophobic region. So not all bacteria have those, but I'll show you some more detail as we go through this. Oh, I guess right now. So the membrane, when you look at our membranes, our cell membranes, there'll be some composition of like sterols that will help strengthen it. And so eukaryotic membranes, like us, we have sterols, including cholesterol. Haploids, though, are what we tend to see in bacteria because they're prokaryotic, remember now. And so at least in the domain bacteria, when they look at their cell membranes, they see these haploids and they chemically and structurally look similar to sterols, but they're not exactly sterols. So they had to give them their own little term, haploids. But basically, sterols in our membranes help strengthen our membrane. And that's what haploids do. They just help strengthen the membrane. And so if you look at a comparison structure here of the chemical, so on the left is a sterol, a basic unit of a sterol in cells like us. And then cholesterol specifically, because most of us have heard that one quite a bit. So you see cholesterol structure, and then the haploid structure that they find in bacteria. So eukaryotic cells don't seem to have haploids. All right, so it seems to be special for the domain bacteria. And here's a close-up showing how they're interspersed in the bilayer. And again, they think that it's there to kind of give a little bit of structural rigidity and support. Okay. Now here's a slide that has a little bit more detail on the glycerol portion, and then what you see here, where they say this R group that you see, those are the fatty acid tails. They just said R because they don't want to put too much on this picture. What they're trying to get you to look at is the linkage where you see ester versus an ether linkage. So if you've had chemistry, they're pointing out the double bond versus single bond, the ester versus the ether. And I know that sounds like a picky detail, but it seems to be a fairly standard thing that we see going on, where if you look at bacterial membranes and eukaryotic membranes, we have ester linkages showing up. But if you look in the prokaryotic cells that belong to the domain archaea, they tend to have an ether. And I know that sounds picky, but it just seems to be a very standard thing. It's very rare to see this rule being violated in any way, right? And so that ether versus the ester. So this is right where the fatty acids connect, okay? It's right there as part of the cell membrane. And so, if you're looking at a newly isolated cell that you don't know what it is, that's one thing you could do. You could ask, does it have an ether or ester linkage? And at least you could narrow it down if it belongs to the domain archaea or into the bacteria. And then this figure C here is the isoprene. It's not showing up there. Oh, yeah, it is. The isoprene unit. So that's the other thing. When you look at, we're going to get into that a little bit. Archaea, they don't have true fatty acids. That's another big difference. All right. So the R group here in the domain bacteria and eukarya are fatty acids that are dangling off. But here, actually, they're isoprene units, which is a little different. So let me show you what I mean by some of the archaea. So here we have some archaea and some funky things going on here. So, well, first of all, you can see they're showing the ether linkage. So right there, that's a tip off. It could be the archaea. And then all of those ziggy-zaggy things, those are isoprene units that are bonded end to end. And what you can see here is this top picture, figure A. You have to imagine now another one of these whole things is right over here, like in a mirror image. So similar to how we have our bilayer membrane, you know, where essentially look at this. This is a mirror image to this, right? So some archaea membranes are similar to our bilayer membrane, where they have that little gap in the middle, and then it's essentially a mirror image. So that's what they're trying to show in figure A there, the phytonol. This is being shown here. Here's the phytonol again, okay? You don't need to know the word phytonol, but if you can understand that some archaea membranes look very similar to our bilayer membranes, except that the tails hanging off there, those are really isoprene units, not true fatty acids. Then the other thing we see with archaea, some archaea membranes, they don't have a little gap. And so it's not a bilayer membrane. They don't have this gap. It's just solid. If you look at figure B here, there is no gap in showing up anywhere in the hydrophobic area. There's nothing. It's just solid. So then they call it a monolayer. It's solid. So if you have a monolayer as opposed to a bilayer, the monolayer, if you look at its structural properties, it's stronger because there's not this little gap here. So it is a stronger membrane. And you tend to see that in bacteria that hang out in some pretty extreme environments where they need to toughen up a little bit to make it through living in that habitat and actually survive it and thrive. And so it's very context driven of where you find these microbes, how they're going to design their membranes. And so the other thing too, back here, you can see that some of these isoprene units do form more complicated structures. And so again, not all archaea look like this, but some do, because it helps them better adapt to their more extreme environment. So very often, if you were to go into a submarine and go deep into the ocean and collected some samples there, you'd probably find a lot of archaea that have a solid monolayer membrane because it helps them deal with the extreme pressure. If you've ever scuba dived, you know the deeper you go, the more pressure your body is under, and you have to be aware of that. And so this helps them deal with that when they get that deep. In fact, the lower layers of the ocean, the archaea win. They're dominant compared to cells that belong to domain bacteria because they have these extra special adaptations. So the cell membrane can vary. Let's see. I don't have a picture of that. Okay. Let's think a little bit about the function. So the cell membrane, yes.
The monolayer, like nothing is transported through the membrane.
Yeah, I don't know a ton about that. I haven't read up a lot on that, but it does seem to slow down transport. And that's one of the things you notice with archaea that live deep in the ocean, like their life cycle is longer. They grow slower because it's harder for them. And that's one reason is the monolayer. It's a little tougher to do rapid transport, do something like that.
The monolayer, that one's not fluid at all, that's solid.
It's not solid, but it's more solid than what you see with the bilayer. Yeah, for sure.
So is this still permeable to this?
Oh yeah, it's still permeable, but it tends to slow down transport. You know, it's just, it's a little tougher, slower to do it. But it's a good trade-off. I mean, if it helps them survive that deep, you know, the pressure down deep, you'll do it, right? And either that or you'll burst and die. So so sometimes same happens with plants. If you see plants living in a real hostile, what we would consider a hostile environment, they tend to grow slower, like deserts, desert plants, they tend to grow slow. And that's one of the trade-offs is that they kind of have to do that to adapt to their environment. So, okay, so we already know from basic biology that the cell membrane is used as a barrier, but it's semi-permeable. It allows things to flow in and out. Great, that makes sense. It's also a place to put your proteins for transport and other things. But because bacteria do not have organelles, they don't have a mitochondria inside themselves or a chloroplast or, well, okay, I'm gonna split hairs here. I'm telling you right now, bacteria do not have organelles. Actually, we're gonna learn a little bit later. They have things that remarkably look like organelles, but microbiologists are loath to say that because that breaks a paradigm and we don't like to do that. Or many scientists don't. I love breaking paradigms. I think it's awesome, but so we still say, oh, we don't have organelles, but I'll show you some exceptions to that. Anyways, let's just say right now, yeah, they don't have any organelle. So that means they're forced, if you think about a mitochondria right now, hopefully you've had some lunch, your mitochondria is cranking along. And that mitochondria does have a membrane that's got all its proteins jammed in there. And it's running a reaction to get you ATP. Well, bacteria don't have mitochondria, so they have to do all of that in their cell membrane. So many of the things that we talk about in like animal and human phys or plant phys, where we talk about mitochondria and chloroplast, if they do anything like that, they have to stuff it all in their cell membrane because they don't have those organelles. So that makes the cell membrane of a bacteria hugely important. It is not just a barrier. It is functionally incredibly important for them. And so if you can, that's why a lot of antibiotics that you might take, they mess with the cell membrane, because once you do that, the bacteria has a much harder time surviving. So the cell membrane, the cell membrane is the site of so many of the physiological reactions that have to take place. This last picture, by the way, we'll get into this a little bit more, but basically this is also trying to get across the point that it's in their cell membrane area where ATP is getting made. And so we've already touched on that already a little bit, but we'll get back into that when we get into physiology. Okay. So cell membrane, super important. So that was kind of a refresher on cell membranes in general, and then some special new information on archaea in particular. Here's a mock-up of a basic comparison of the cell wall of bacteria. So now we're in the domain bacteria, and we're looking at the cell wall. So bacteria have cell walls. We don't. And one of the main portions of the cell wall is peptidoglycan. Peptidoglycan. And you see here that there's two comparisons here, gram-positive versus gram-negative. So we categorize bacteria into those two categories mostly. Most of them fit into one of those. And really, it's all based on what is their cell wall like. That's what the comparison is. So it's named after Dr. Graham, who came up with a way to stain them in the lab so that you can clearly tell the difference of what type of cell wall do they have. And then he named the one that has a lot of peptidoglycan, the gram-positive type, and the one that has very little peptidoglycan, the gram-negative type. Okay, so we'll learn about staining this week. And so it's about 90% of the cell wall. When you look at a gram-positive, about 90% of the cell wall is made up of peptidoglycan. So it's very heavy peptidoglycan. But with the gram-negative, you see a little extra stuff going on here. You see very thin peptidoglycan. So only about 10% of their cell wall is peptidoglycan. Then the other thing you see here is this space where the peptidoglycan is located. That's called the periplasm. That's just the physical space between the cell membrane. And then this blue wavy thing that they've drawn, that is actually part of the cell wall, even though they call it a membrane. It's a little weird. But it's the outer membrane of a gram-negative cell wall. So we just kind of lump it in as it's cell wall structure, even though it's a membrane, but it's a funky membrane. It's not like this typical bilayer membrane, and we'll get into that. Okay, so when you do the gram stain, this is what you're gonna be keying in on. The stain is gonna pick up if it's, has that really thick peptidoglycan or a much thinner. And because of that thickness, you can stain, you can do this differential stain technique. And then visually you can tell if it's gram positive or gram negative by the two different colors that show up on your slide. So speaking of the gram stain, you're supposed to do that tomorrow. One of the autoclaves failed. And we had a batch of our test tubes filled with broth that we grow the bacteria in. Went through that cycle, and when it came out, it looked perfect. After a couple of days, however, it became contaminated. We didn't find that out till after we inoculated them with the bacteria we want you to work with. So now we have all these contaminated tubes that are useless for tomorrow. So I'm trying to figure out if I can sub in some certain other bacteria for you. But I wanted to let you know that we may have to delay the gram stain until maybe Thursday. Yeah, so I notified our Jing, who needs to keep on top of calling in people to look at the autoclave, but we had a massive contamination because the autoclave didn't work. The right one. I've had problems with both over the years, but. So I asked Dr. Sharp if he had any microbes up and running we could borrow, but if he says no, then that's fine. We'll just shift. So just be aware, tomorrow's lab, I'm gonna talk with Duncan and Isabella right after class here, and we're gonna figure something out. So you will do something with microscopes and looking at slides. I just don't know exactly what yet. We're gonna talk about it. So the joys of working with living microbes, failing autoclaves. Those autoclaves have failed us more than once. So, but we will definitely do the gram stain. This is like a huge thing that you have to learn. If you walk out of a microbiology class in college and never learn this, it would be terrible. So you will learn this. Okay, it's just probably not tomorrow. All right, and then this is where you can draw the line. So through this slide will be on the first exam. Okay, everything through this slide. And then the next slide, I'm going to show you, this will not be on the exam. All right. So this is more detailed of what's going on in this slide. So I'm just going to give you the introduction here. That will be on the exam. And then the next exam, you're going to be learning more about the detail of it. Okay. So I'm going to mention a few other things on the cell structure, and then I'm gonna leave a few minutes for the end so that we can talk a little bit more about the exam, so that you're feeling good about approaching that. All right, so here's a mock-up of the cell wall. A lot of books will call it the cell envelope, which is fine. I learned it cell wall, so that's how I tend to speak about it. Here they actually have electron microscope pictures showing the difference. And those are nice pictures. And then you see here, if you compare the left versus the right, you can see quite a bit of difference here. And so the purple and blue here, we're getting into the peptidoglycan. So I wanted to get into that a little bit. And so they're trying to show that there's many layers of these molecules that are in these long chains, many, many layers. So that's why the gram positive it has such a thick peptidoglycan. And then with the gram negative, you can see it's pretty much a single layer. And then there are some bacteria. Here again, there's a gram positive, and they're trying to show this big purple structure, that's the peptidoglycan, and then here you see the peptidoglycan much smaller, and then this funky outer membrane. And then acid fast, we're gonna learn that stain too, and that they have different cell wall. I'm not gonna go into huge detail. You'll learn more about it in lab, but basically the acid fast bacteria have a lot of interesting lipids that are stuck to their peptidoglycan area, and it makes them look, when you do the acid fast stain, you can clearly pick up on them, which you'll probably do on Thursday. So I like this picture because it's trying to get across that even though microbes are tiny, tiny, tiny things, they are doing so much on a minute by minute basis. They are chock full of proteins and enzymes, DNA, plasmid, and then their cell membrane as well is chock full of proteins and so much going on, not just, yeah, so here they have the ATP synthase, so they're trying to show there's lots of ATP synthases, so you can make ATP. And then you have other proteins embedded in there too. So it's really a workhorse area for the bacteria. But let's go beyond the cell membrane out into the cell wall. And I wanted to talk about that to some level of detail. So when you look at the peptidoglycan, it's essentially two sugar derivatives. One is called N-acetylglucosamine, which you can abbreviate down to NAG, N-A-G. The other one is N-acetylmuramic acid, which you can abbreviate down to NAM. And if I ask you a quiz question or whatever, you can just put NAG or NAM. You don't have to spell it all out. Okay. But so you have the NAG and NAM sugar derivatives, and they bond with each other, and then they alternate down that long stretch. So it's NAG-NAM, 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 and they form these long chains that then get kind of stacked up for the gram-positive anyways into that thick peptidoglycan. So then that's not the only thing though. You have that, but then you also have amino acids attached to the NAG and NAM. And depending on which microbe you are, you're going to have L-alanine, D-glutamic acid, and then you'll either have diaminophenolic acid, which abbreviates down to DAP, in mostly gram-negative type bacteria, but instead gram-positives, replace that with L-lysine. And then you have a D-alanine. So L-alanine, a D-alanine, glutamic acid, they all have that. But then depending on if they're gram-negative or gram-positive, they might have DAP or L-lysine. Now, those amino acids, so there's four. Okay, you have the L-alanine, D-glutamic acid, one of these, and then D-alanine. So there's four amino acids that hook on to one of the sugar derivatives. So 4 means tetra. So they often refer to it as a glycan tetrapeptide. Glycan is the sugar derivative. Tetra means 4 amino acids. Peptides are the amino acids in short chains. Okay, so often you'll hear it described that way. Peptidoglycan is a glycan tetrapeptide. So if you look at the structure more carefully now with the sugar derivatives and then the amino acids, you would see something like this, where you have chains of peptidoglycan. So the hexagons are the sugar derivative. And you can see they've chosen blue for the NAG and purple for the NAM. And then coming off of only the NAM, you see those brightly colored balls. Those are the amino acids. And it allows those amino acids then hook up to other amino acids coming off the nearby strand. So that's like an like a connection between the sheets of peptidoglycan, so they don't like fall apart from each other. Right, and so it's a way for them to stay cohesive to their neighboring chains. If that makes sense. Okay. So here's a blow up of the 4 amino acids of one, and then it hooks up into the 4 amino acids of the neighbor chain. And that's just to help stabilize the layers or the sheets of peptidoglycan. So if you, here's another image of that. And so this is a good way to think about it. Peptide cross-links. They're cross-linking the neighboring NAGNAM chains, okay? And it's basically just so that all those chains of peptidoglycan, especially in the gram-positive, they can stay together. They can be cohesive and not fall apart from each other. So the NAG and NAM, they're showing this beta-1,4 linkage. And then they're saying a beta-1,4 linkage is lysozyme sensitive. So the reason they're pointing that out is that they know that a lot of microbes that coexist with, like, with us, because they have that beta-1,4, we have lysozyme, we have that enzyme. And we have it in our saliva, we have it in our tears, a few other places. So if you eat something, some kind of bacteria, the lysozyme immediately starts attacking the beta-1,4. And so if you can break down their cell wall, you go a long way toward killing it. And so that's why they're pointing that out. And then there are some other microbes, like many in the domain Archaea. They don't have this kind of linkage, but that's okay because we don't eat or interact with Archaea in that way, like we do with other bacteria that might make us sick if we eat or drink contaminated food. All right, what else? Oh, so here, if we go back to this one, if you look at figure B here, I don't know if this helps or not, but if you know what I mean when I say chainmail, like the knights in armor, and they have to wear that chainmail, that's how they often describe the overall configuration of the cell wall, where you have that chainmail-like appearance of the strands of the sugar derivatives with the amino acids cross-linking like that. Yes, I'll show a picture to try to get at that. It's a little hard initially when you're learning this. It's, unless you have good spatial reasoning, it's a little, at least for me, it was hard to learn this. But I put in some extra pictures in a minute here that will show that. So this is some of the way they describe it in words. They'll say it's like a sheet-like configuration of strands or chains of peptidoglycan that are neighboring each other, laying adjacent, and then they're cross-linked to each other. So this is a good way to think about it in words. Okay, and then there's a little bit of difference here with gram negatives versus gram positive in one of the choices of the amino acids, probably 4. And then I'm going to show you a picture here where gram positives sometimes put in a little added twist where they have an interbridge of some amino acids, and I'll show that to you in a sec here. And then the gram negatives tend to have like just one sheet of peptidoglycan. And then the gram positive tend to have many layered and stacked and wrapped around the cell. Okay, so this is all in the PowerPoint. You don't have to write all this down. So here is comparison of a gram negative versus a gram positive. So E. coli is gram negative. They're still saying, yep, you have NAGNAM, 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 laid down in a chain, alternating like that. You have these cross linkages with the 4 peptides linking to each other. And then here they're showing the interbridge. Some gram positives will throw in a little extra spice and put some, glycine is often a favorite one because it's a small amino acid. So they'll go ahead and stick some glycine in there as part of the cross-link. And then this picture down here, they're just trying to show again with a gram positive how you have multiple sheets like that. And again, it kind of looks like chainmail, if that helps think about it at all. Here's another image, again, just showing pattern. And then here they're trying to show kind of how it wraps around. Oh, and then the other thing too, especially with gram positives, they see glycolic acid quite a bit, which is embedded into the thick sheets of peptidoglycan. And they either call it teichoic acid if they're just teichoic, or if they're attached all the way down to where the lipid, see all the way down there, it's interacting with the lipid of the membrane. Then they call it a lipoteichoic acid. So that's the only difference. How long does it stretch down? There's a bigger image of that. All right. So gram positives, many, many layers of the peptidoglycan. Gram negatives, very slender, but they do have that outer membrane. And it's funky. They refer to it as the lipopolysaccharide, which abbreviates down to LPS. And if you've had Med Micro, I'm sure that should ring a bell. LPS, or the lipopolysaccharide. Okay. There's also that periplasm, which is just a space. It's a physical space that's between the cell membrane and the outer membrane. And the peptidoglycan is stuffed in the periplasm. So this outer membrane is not like the cell membrane, especially when they noticed that it doesn't seem to have any selective permeable capabilities, or not much. It allows a lot of stuff to go through. So that outer membrane isn't really the selective barrier like the cell membrane is. And when they look inside the periplasm, that space, yes, the peptidoglycan is there, but they see a ton of other stuff in there. Lots of proteins and enzymes. It's kind of like this superhighway system area where you get a lot of activity going on, because that's like the last stretch of space between before you hit the cell membrane. So here's a gram negative, pretty busy slide. So starting at the bottom, you see the cell membrane, lipid bilayer, phospholipid bilayer membrane. Then you see a relatively thin peptidoglycan in the periplasm. Then this is your outer membrane. You can see it's a bilayer. It is a bilayer because you have that gap, but they're not mirror images of each other, like what you see in the cell membrane. This outer portion of the outer membrane is very different. And that's where you see the lipopolysaccharide, the LPS. So these different colored hexagons like that are again the sugar derivatives that we'll be talking about. Okay, so that is very different compared to what we just saw with that. And so even though they're tiny, they can look very, very different and behave differently because of the difference in their cell wall. This will come back. We'll talk more and more about the LPS, especially as we get into human microbe interactions. We need to think about the LPS during those concepts as well. Any questions? Yes. Yeah. Yeah. Oh, no. How it's different here, you mean?
Yeah, just like the way I was looking at it, like it might be similar amount of targets that involve intermolecular interaction.
Oh, yeah, I don't know. That's a good question. I mean, they almost drew it here like there's something going on, you know? But yeah, I'm not sure. It's a good question. I do know they actually say that having that monolayer, yeah, often use the They say it keeps the cell membrane from peeling apart from each other, like those fatty acids. So. Any other questions? All right, what about the exam? So let's spend a few minutes talking about that. Today's Monday. Yes, just up to that one slide though, because this is a big topic. So you don't have time to learn it all for sure. So even what I covered today, we're not going all the way through that. We're stopping here. Okay, so, so going back, oh, let me pull this up here. Hang on. So I have a document